C means G, C means G. Okay, so I'm okay. These are all small RNAs. Okay, these are all small RNAs. Okay, I am coming outside in a lab, outside the cell. I am synthesizing these small RNAs. Okay, so ஒரு செல்லுக்குள்ள இந்த mRNA form ஆயிடுச்சு. This is going to be converted into a protein, but I don't want that to happen. So what I am doing is I am manufacturing a small siRNAs. Okay, this is called small interference RNAs, and these RNAs are complementary to this. இப்போ நான் இத I am injecting into the cell. அப்படினா என்ன ஆகும்? இதெல்லாம் போய் இது கூட பைண்ட் ஆயிடுமா? இது போய் இது கூட பைண்ட் ஆயிடுமா? Right? I will write it separately. Okay, I am labல நான் synthesize பண்றேன் SIR என்னது? A U க்கு பதில் sorry A க்கு பதில் T, T க்கு பதில் A, G க்கு பதில் C, G, G, G. These are all the 3 siRNA நான் synthesize பண்ணிட்டேன் labல. I have synthesized. Okay, now I am going to inject this into the cell. அப்படினா என்ன ஆகும்? இது அப்படியே மூவ் பண்ணி வந்து this will come and bind here. This will come and bind here. This will come and bind here. ஏனா I said always A will pair with T or U, G will pair with C. Yeah, so they will have a natural affinity. They will come and bind. Once this comes and bind, இது bind ஆயிடுச்சுனா that's it. Further protein synthesis will not happen. இது single strand இருந்தாதான் உங்களுக்கு ribosomes வந்து bind ஆகி உங்களுக்கு amino acid synthesis amino acids எல்லாம் join பண்ணி protein ஆ மாறும். But once இதுல போயிட்டு இந்த small RNAs போய் இதுங்க கூட complementary ஆ bind ஆயிட்டா that's it. Further protein synthesis is stopped. Okay, so this process is called as RNA interference. What you are doing simple, as the name indicates, you are interfering with this RNA. You are interfering in this RNA and stopping it for its further translation into protein. You are not allowing it. How you are not allowing it? You are synthesizing small siRNAs and injecting it into the cell and making it to bind to this mRNA. Thereby this mRNA will become these are all non-functional. Okay, this will not be converted into protein. Okay, so by this way we are stopping the gene expression, right? Okay, say for example, I said in sickle cell anemia, hemoglobin malformed hemoglobin it should be disc shaped. Hemoglobin இப்படி இருக்கணும். ஓகேங்களா? But sickle cell பாத்தீங்கன்னா it will be like this. Okay, so I don't want this hemoglobin to be produced. Okay, this is dangerous for me. I don't want this to be produced. I want to silence this. How can I silence this? இப்போ இதுதான் hemoglobin gene. Hemoglobin shortly we call it as HB gene. Okay, this HB gene will become HB mRNA. Then இத நான் protein-ஆ convert பண்ண விட்டேன்னா it will become sickle cell. For the affected for normal individuals it will be normal, but for sickle cell anemia persons this will be sickle. I don't want this to happen. So what I can do? I can simply inject these siRNAs, these small RNAs. இதுக்கு complementary இருக்கிற small RNAs நான் inject பண்ணேன்னா so they all will go and bind with this fellow and thereby that's it. It becomes non-functional. Further hemoglobin is not produced. Thereby I am silencing the gene. Okay, so RNA interference is a mechanism to silence the gene. Okay, as I said, genetic engineering is not only to add a gene, it is also to silence a gene, and one such mechanism is RNA interference. Okay, fine. Now let us see the previous year question so that you will have. Okay, so this question, so let me read it. RNA interference technology, RNAi technology has gained popularity in the last year. Why? So நம்ம ஒரு example பார்ப்போம். Here as I said, first choice, it is used to develop gene silencing therapies. Yes, I said so I don't want hemoglobin sickle cell anemia hemoglobin வேணாம்னா if I அது complement ஆனா கூட gene அது silence பண்ணிடும். Yes. Then choice statement 2, it is used for developing therapies for the treatment of cancer. Yes, exactly. Why cancer is happening? Cancer is happening due to rapid proliferation of cell. Cancer cell for example, okay, I'll use specific example. For example, breast cancer. Yeah, so women are facing breast cancer. In breast cancer, for example, this gene BRAC2 some gene is there. This gene is overexpressed. Overexpressed na என்ன? DNA ல இருந்து many mRNAs are formed. Many mRNAs are formed and all these mRNAs are converted into protein, and this is responsible for causing the breast cancer. Okay. Now, how to arrest this? In the mRNA, I don't want this to be converted into this protein. So, as I said, I can make a small siRNAs which are complementary to these mRNAs. Okay. I inject the siRNA ellam poitu idu kuda bind aayittu further conversion of further translation into protein is not possible. So, thereby this RNAi technology I am also using it for cancer therapy. Then the third thing, it is used to develop hormone replacement therapy. That is wrong. Hormone replacement therapy is totally a different concept where you are injected with hormone, where you are injected with hormones like progesterone or estrogen. So that is totally a different concept. Here that doesn't play a role. Then third thing, it can be used to produce crop plants that are resistant to viral pathogens. Exactly. Okay, ma? How this RNAi technology is used to produce a viral resistant plant? Say for example, I have a plant. Okay, so I have a plant. Okay, this is a tomato plant. Okay, this is a tomato plant. This tomato plant is often infected by a virus called TOLCV, tomato leaf curl virus. Okay, this virus often infects this, and it causes the curling of leaf, and it is loss for the farmer. There will be no yield and other thing. Now I want to stop this virus from infecting my tomato plant. Okay, so what can I do is I can, in the say here you understand how genetic engineering comes. So now previous examples, so you take you synthesize siRNAs, the small interference RNAs synthesize panni, you inject. Okay, I said you na ipa hemoglobin I don't sickle cell hemoglobin hemoglobin so I can synthesize a complementary RNA. If I inject into the cell, that will go and bind to the mRNA and it will stop. But injecting into each and every cell is not possible. Instead, enoda cell and the small siRNA produce panna, that will be very functional. Yeah, so that I can do. What I can do is I can change this gene. Say for example, in the plant or chromosome, tomato or chromosome, or chromosome. Okay, so tomato or idu or chromosome. What I can do is I can add this S and the small interference RNA produced pandra madri gene and I will add panilla. Okay, so you can add the gene that produced this small interference RNA. So in the tomato or in the chromosome I add panita. So all the cells in this tomato plant will carry this gene. Okay, so in the siRNA against this virus, I know this in the virus oda RNA eppadi irukunnu enakku theriyum. I have studied. Okay, we have the sequence of this RNA. So based on this sequence of the RNA, I can synthesize a gene for siRNA and I can incorporate into this tomato plant. Okay, so this tomato plant, just like vaccination madri da, so this tomato plant will be producing siRNA for against the mRNA of this virus, this tomato leaf curl virus. Okay, fine. This is gene we introduce natural by transgenic method. We are introducing this. We'll see. By transgenic method, we are introducing into this plant. So all the cells or the leaf, stem, everything will be producing this siRNA and will be waiting. Okay. Now one fine day, this virus. Okay. Say this is that virus. This virus is coming and infecting this plant, this tomato plant, and it will inject its genetic material and it will synthesize the mRNA. Now what will happen? Already this tomato plant is having the siRNAs complementary to this mRNA. All the siRNA will come and bind to this and it will chop this. That's it. So your virus is dead. Okay. So thereby only if this mRNA is converted into protein, this virus can survive. It can replicate and it can become manifold. Even the COVID pathina, it is entering, it is binding to our cell surface. It is entering, it is hijacking our genome, it is replicating, then cell damage pannittu oru coronavirus villa pochuna, you are having hundreds of coronavirus. The mRNA is converted into protein and this. But here what we are doing is already the gene to produce this siRNA is injected into, is there in this plant, and it has already synthesized, it is ready with that siRNA. So whenever this virus invades this plant, this mRNA will be, when it produces the mRNA, the siRNAs will bind to this mRNA and it will be chopped so that no further reproduction of this virus. Okay, here the one concept I have used the word chop. Why I use the word chop is, so I don't want to go deep into the mechanism, but just know this. I said you know, so this is mRNA and you are making siRNA and putting it. So the siRNA will come and bind to this. Okay, so inside the cell there is a mechanism called, whenever an mRNA and siRNA is bound, okay, whenever they are binding, then the cell will process this mRNA for degradation. Various enzymes will come and chop this into pieces. Okay, the similar fate will happen for the virus. In case in our tomato virus, where we have genetically modified the tomato plant to produce this small interference RNA against the virus. So it is ready with the siRNA. Now the virus is coming and invading. The virus is making the mRNA. Now this siRNAs and mRNAs will bind. That's it. The cell will drive it for chopping. The protein further, the virus cannot replicate and it is dead. Okay, so that is the statement. The statement 4, it can be used to produce crop plants that are resistant to viral pathogens. So the answer is 1, 2 and 4. Hope you understood the meaning. You should understand. So it is used for gene silencing, yes, in statement 1. It is used for treating cancer treatment. Yes, cancer is due to rapid proliferation of a particular protein or hormone or enzymes. So here, as I said, in breast cancer, that particular protein is expressed more. So I am putting an siRNA and I am stopping that gene. Thereby I'm getting rid of cancer. Okay, for in plants against viral infection. Okay, this is RNA interference technology, RNAi. Now the next thing is CRISPR Cas9. This is also a Nobel winning concept. If I'm right, it is 2019 or 20. Please go through. This is also a Nobel winning concept. Let us not go deep into the mechanism. Let us see what this CRISPR Cas9 system is. As I said, that RNAi system is for gene silencing. It is to knock a gene. It is used to silence a gene. This CRISPR-Cas9 system is a dual purpose. Here you can silence a gene, also you can express a new gene. Now, genetic engineering purpose is you have to add a new gene or you have to knock out a particular gene, right? So in CRISPR-Cas9 we are going to INN pakla. So nothing but Cas9 is a protein. Okay, Cas9 is a protein. Okay, Cas9, this is a protein. This is Cas9. In the Cas9 protein, you are keeping one mRNA called as guide RNA, not mRNA. This is called as guide RNA. In the Cas9 protein, I am keeping one RNA called as guide RNA. If okay, I am keeping a guide RNA. Okay. So what is this CRISPR system is? I am putting it in a very simple way. Say for example, this is your DNA. Okay, this is your DNA, Naka. Okay, okay. This is your DNA. This is your CRISPR system. This Cas9 protein having a guide mRNA. So in the mRNA, complement. See this strand is having. Okay, I put like this. This DNA. Okay, this strand. ATGC. Okay, here it will be. For A it will be U, for T it will be A, for G it will be okay. This is guide RNA. Now what it will do? This Cas9 protein having this guide RNA will come here. Okay, it will slowly move and it will come and sit here. Okay, so it will come and sit here, and this guide RNA will also sit. This guide RNA is having U, A, C, G. Right? Now these two are complementary. I said, whenever these two are complementary, these two will bind. Okay, these two has power. Now, just now I said, whenever in a madri or in the RNA in the hybrid structure form, then the cell will come and chop it. Okay, now this system will cut it. Thereby, this particular gene is knocked out. Okay, so the Cas9 system I'm telling, this is a Cas9 protein. Inside the Cas9 protein, I am keeping one. I am synthesizing one RNA and I am keeping it as guide RNA. Okay. Now this is the DNA. Now this particular gene, say I am telling, let us take one example itself, insulin gene. Now this insulin gene, I want to knock it. Okay, so I want to knock this insulin gene. So what I can do, I can just push this will come and sit here, and this guide RNA will sit here. They both will form a pair. Once they both forms a pair, our cell system will come and recognize and will chop this. Thereby, this insulin gene is chopped away. Okay, so by this way, okay, அந்த ஜீன் முன்னாடி எப்படி இருந்தது, இப்படி இங்க இன்சுலின் ஜீனோட இருந்தது. பட் இது அப்படியே வந்து இதுல உட்கார்ந்து இதுவும் பேராஃபார்ம் ஆகி சாப் பண்ணதுக்கு அப்புறம் this insulin gene is removed. Thereby I am silencing this gene. Okay, this is CRISPR-Cas9 system. Okay, then how I said it is not only used to silence, it is also used to synthesize new gene. Now ஒரு புதுசா ஒரு addition of gene கூட நம்ம இத பண்ணலாம்னு சொன்னா எப்படி பண்ணலாம்னா, see once this is இங்க break வந்துருச்சு இல்லையா, so this is break வந்து உங்க செல் சிஸ்டம் ஓகேவா பைண்ட் ஆச்சு. பைண்ட் ஆனதுக்கு அப்புறம் once அந்த டபுள் அந்த ஹைபிரிட் ஸ்ட்ரக்சர் ஃபார்ம் பண்ணோனே your cell system will recognize and come and chop it. Once chopped, your cells will start synthesizing it. New system, your cells will start synthesizing it. Okay, and while synthesizing, in this guide RNA, if you add your desired, say for example, already in the insulin irundhudu, okay, na adha replace pannittu, say I want lactase gene அப்படினா, I can add this lactase gene to this guide RNA. I can add this lactase gene to this guide RNA. Okay, now this insulin gene will be chopped and this lactase gene will be newly added to this genome. Okay, so this is a very complex mechanism. I have put it in a very simple way and in a very crude way to help non-biology students to understand. So just have some knowledge. So what is this CRISPR-Cas? Why it is called as CRISPR? All this. So this is a natural system found in bacteria. Okay, have some basic knowledge. This is a natural system found in bacteria. So bacteria uses this system to prevent them from the invading viruses. You all know, right? Bacteria is infected by a virus viruses called as bacteriophages. Bacteriophages are viruses. Okay, you would have studied in your smaller classes. This is bacteriophage. So this will come and this is bacteria. This will come and infect this virus. Adoda genome idhula insert panni adu replicate aayittu and it will kill this virus and it will reproduce. That is killing this virus. To escape this system, okay, to escape this system, these bacteria are having a system called as CRISPR-Cas9. In the CRISPR-Cas9 bulima, the invading viral genome, they are chopping it. Okay, a similar mechanism. They will be having this Cas9 protein and have this Cas9 protein will have a RNA. That RNA will go and bind complementary to this viral RNA. They both will bind and it will be chopped. Okay, so it is a natural system found in bacteria, and now we have modified it and we are using it for our genetic engineering purpose. Okay, so what I have said is a very crude knowledge. Don't be. This is not the exact system. I have simplified it to the core so that you can understand. Okay, so what is CRISPR system? So this is, see, you can see here. This is the Cas9 protein. This Cas9 protein will have a guide mRNA. This green color, this is the guide mRNA. So that will go and bind complementary to this DNA, and that DNA is chopped away. Okay. Then this question: what is Cas9 protein that is often mentioned in news? That's the statement 1, option A. Cas9 protein, molecular scissor used in targeted gene editing. Yes, you can edit the gene. I said you na specific mRNA adla guide RNA vechittu DNA la poi bind aayittu it can edit, it can go and make a chop, and after that you can add your desired gene. So it is a molecular scissor used for targeted gene editing. It is a biosensor, pest resistant, all that are irrelevant option. Your option 1 will make it clear. Okay, so this is about CRISPR-Cas9 system. Okay, now with this Now let us go in depth into genetic engineering part, how this transitioning plants and animals are made. Let us see that in the next video. Okay, thank you.